Thanks everyone for being here. This is our error-corrected sequencing meeting for our Emerging Systems Toxicology Committee. Will be our annual meeting. I know quite a few of you were on our meeting yesterday for carcinogenomics, but if not, I'm going to go through a similar quick intro, and then the rest of today will really be focused on error-corrected sequencing and all the progress this year. Okay, so I think most folks know, but this is a HESI initiative, the Health and Environmental Sciences Institute. We bring together experts from the different sectors, industry, academia, and government, and other research institutes to work on issues you couldn't solve just in one lab or one sector, mostly focused on toxicology and assay development. And I think I know most folks, but if you don't, I'm Connie Mitchell, Senior Scientific Program Manager, and myself and Chrissy manage our Emerging Systems Toxicology Committee. And this really houses our omics work, but can be a little bit broader and deal with systems toxicology as well. And so we have a number of projects under this committee, but we're also made up of the experts, kind of what I just enunciated with the different sectors being involved. So for this committee, we have really good public sector endorsement, commitment, experts working. And then we also have really good government as well, mostly from North America and Europe. And we also have some other nonprofits involved in some of our projects. Just gonna, mute a few folks. We also have really strong private sector participation. So we have mostly folks from ag chem, from pharmaceuticals, but also from other sectors like industrial and really some of the biotech sectors that help us with the assays and the technology themselves. So really huge thanks to all of you for being involved. We have 6 different projects. Today we're going to hear about error-corrected sequencing. Yesterday we had our meeting for carcinogenomics that went really well. And each of these groups are going to have their own meeting, with the exception of POD that just had a workshop. So they'll put out a workshop report in the next couple of weeks. Otherwise, we have some other ones upcoming in the month of November and one in early December just to finish up some lab work. So look for those meetings, and if you don't have them on your calendar, let me or Chrissy know. Just one note, for all the work we do at HESI, we're really committed to making it open access. So for eSTAR, our Emerging Systems Committee, we've had a few papers come out recently, not for error-corrected sequencing. We hope to have some publications next year, but for eSTAR generally. So we had one that followed up from an SOT report talking about these shorter-term mechanistic studies in animals and how they could be a bridge. So I think some of the work today will apply. We also had the OASIS, which is our cell painting project. Their strategy paper just came out. And then also one of our long-standing projects, TGX-DDI, our biomarker, they just had their ring trial published. So a lot of work has come out just in the last month or so. So just wanted to make folks as aware as we're not having the bigger annual meeting this year. And just in terms of outreach, you're going to hear about the outreach we did in 2025 for this group. But looking forward, at least staff, myself and Chrissy, will be at SOT and the ICEM meeting, and we're also open to other opportunities to highlight the work from this project and for others. So with that, today, this is a Teams meeting. It's not a webinar. We want folks engaged. We want people to be able to ask questions and be able to be part of the conversation. So there's multiple ways to do that. I'm sure most folks know, but you can chat throughout the day. You can raise your hand. And then if something really resonates with you, you can also use the React. So I think with that, I'll move to our error-corrected specific slides, and we'll get kicked off today. And so huge thanks, the whole team that does this. A lot of thanks to Patricia and to Megan for leading this team. And you'll hear also from James, who's done a lot of the lab work and really support for this project. So what we're going to do today is give an overview of the group objectives. We'll give status updates on the foundational studies, and then we'll discuss what we want to do next year. And I'll go through this, but please, Patricia, if there's anything you want to add into, feel free as we go. But basically, our working group objective is to evaluate error-corrected sequencing technologies as a potential tool to measure clonal expansion that could contribute could contribute as an early tumorigenicity biomarker. So can we use clonal expansion in these tools in concert with other tools to look for tumorigenicity? And we came up with this framework to how can we actually move from having a technology we're interested in to actually being, more comfortable with it. And so the first thing we identified is we have the technology, we have the panel of genes we want to look at. We'd like to run additional compounds and studies. So that's really been our focus the last year and a half, is trying to find studies that already exist, samples that already exist that we can utilize with the technology we have. There might be some other finessing to genes, working to establish thresholds. If we see a clone, what does that mean? And then moving towards regulatory acceptance is a later goal, but really laying that foundation. So as part of that foundation, really you all helped us create an inventory of samples we could potentially use. And we've identified 2 to start with. This is really to help us get going and what we're going to hear about today. There's other ones on our list that are potentially available we could use and look at, but these are the ones we've gotten started with. So I think with that, James, I'm okay to switch to you unless Patricia, there's anything you'd like to add in terms of background.
No. I think okay. Thank you.
Okay, James, I'm happy if you'd like to share because I know you have some animations.
Yep. Thank you for the opportunity to present our results here. This has kept me busy the past year or so. I think I'm sharing my screen, right?
Yeah, we see your screen. You're not in presenter mode yet.
Okay, there you go. Okay, so hi everyone. I'm James Alcaraz, and I'm a postdoc here at the University of Ottawa. I work closely with the leadership of the ECNGS committee to help lead and coordinate our first studies under this initiative. So really my role focuses on supporting the design, implementation, and analysis, helping move our research from concept to data. So just to remind everyone on the background while we're doing this pilot study, we're really still at the very beginning in terms of the feasibility of using clonal expansion as an early biomarker of carcinogenicity. Our first study deals with determining the appropriate sampling size for detecting clonal expansion. So what we really wanted is to determine whether there will be differences in detection of clonal expansion if we use multiple small punches versus a large tissue homogenate. They have their own advantages and disadvantages. Small punches would amplify the detection of large clones, but it requires an assumption that there is an equal chance of punching tissues with cancer driver gene mutations. On the other hand, using large tissue homogenate may manifest diminished levels of individual mutations due to signal dilution, but it may also concurrently present a greater diversity of distinct mutational events. For this pilot study, we used samples from a non-genotoxic imidazolidine analog. This compound has been tested, and it showed that it's negative in a battery of genotox tests, but has been shown to activate the CAR mechanism. In a 2-year rodent cancer bioassay, it caused hepatocellular carcinoma in male mice, really making it a strong test case for whether ECNGS, in this case, duplex sequencing, whether it can detect early tumorigenic changes. So we focused here on male mice from a 90-day parallel study comparing the controls versus the exposed group. The exposed group is exposed to 114 milligram per kilogram per day, which is a concentration that's higher than the concentration where they found hepatocellular carcinoma in the 2-year study. Overall, we have 3 main objectives for this pilot study. First is to test whether duplex sequencing can really detect mutations in the system. Second is to establish reliable sample preparation and sequencing parameters. And lastly, to compare tissue punches with homogenates to see which is more effective for detecting early clonal expansion. We have here 10 mice per treatment group. These samples were donated by our colleagues from Syngenta. Here at U Ottawa, we extracted the DNA either from a large tissue homogenate or from 3 small punches. HESI provided the duplex-seq library prep kit which we use to prepare libraries using a target or a targeted panel of 27 cancer driver genes. Merck then sequenced these libraries at very high depth using a NovaSeq instrument. Finally, we assembled a sub-working group to develop the analysis pipeline, primarily led by Paige and Megan from FDA NCTR with us here in U Ottawa and the Health Canada. So these are our results from the pilot study. This figure shows mutation burdens across all samples. So the data used here includes singlets and multiplets. Just to orient you quickly on how to read this data set, each of the bubble plot here represents one sample. Each of the bubble is a unique mutation. The bubble size reflects the clone size, and the color corresponds to the gene. You'll also notice here that the punches appear to be bigger. That's simply because each of the bubble plot is for the punches is an aggregate of 3 punches, each sequenced as deep as a homogeneous. So this does not reflect a true biological difference, but overall, this result demonstrates that technology works. Duplex-seq reliably captures early cancer driver gene mutations. So we're still talking about mutations here. On clonal expansion, we defined clonal expansion events or their occurrence based on the depth or the number of alternative alleles. For this analysis, we required at least 2 supporting alternative allele depth before calling it a clonal expansion event. For our data analysis, we developed the workflows in a two-tiered approach. We first focused on using a single statistic. So first, we only looked at the number of clonal expansion events. But that means we're ignoring how big the clones actually are. Another iteration of this is looking at the size of the clone. So we looked at only the mutant fractions. But that means we're ignoring how many clone expansion events are occurring. So what we did was we combined both the number of event counts, or we look at the combination of the event counts with mean clone size. So this allowed us to distinguish between the variables that would affect the number of clones versus those that would affect the growth dynamics or the clone size, and the combination of both. The data analysis has become really complicated. So to do this, we use a Bayesian multi-level regression framework implemented in BRMS. I'd really like to acknowledge Andrew Williams from Health Canada here, who's really the brains of this highly technical statistical analysis. So simply put, we were able to model the number of clonal expansion events and mean clone size together, and we included random intercepts for each animal to capture inter-animal variability. So what we found here is that first, there's a substantial variability between animals, both in the number of clonal expansion events and the mean clone size. And this is true even after accounting for whether the sample was coming from punches or homogeneous, or whether they're coming from the treated group or the control group. We also observed a moderate negative correlation in the number of clonal expansion events and average clone size. The downward trend here shows that the animals with more clonal expansion events tend to have smaller clones, while those with fewer events tend to have larger clones. Now, for modeling the event counts and mean clone size, we found no credible evidence of differences in the number of clonal expansion events, whether comparing treated and control groups or punches and homogeneous and their interaction. For the size of the clones, the vehicle control group showed somewhat larger clones compared to the treated, However, when we broke down by sample type between punches and homogenates, there seems to be little to no credible evidence of treatment effects. So since the previous model showed no credible effects of either treatment or sample type, we simplified the model to focus only on sample type. The idea was to really ask more directly whether punches and homogenates differ in capturing clonal expansion events. So when comparing punches and homogenates, there was no credible evidence of differences in their number of mutation events or average clone size. So our results based on this pilot data set, um, it, basically it shows that there's equivalent information, or the information is equivalent about mutation counts and clone size, whether we use homogenates and uh, punches. And it may or may not be specific to data sets. I don't know. Just to sum this up quickly, we in this pilot study, we've shown that duplex-seq protocols are robust and transferable from detecting mutagenesis in neutral targets, the way they use this in the mutagenesis panel, to really detecting cancer driver gene mutations. The results also suggest that either large tissue homogenate or multiple punches can be used for this type of analysis. And looking ahead, we plan to refine the bioinformatics pipeline and statistical models, conduct power analysis, and hopefully wrap up this pilot study soon. The other study that we conducted was using benzobifluoranthine as
James, maybe we can stop here and see if people have questions about that study first.
Yeah.
If that's okay. And then we can go to the next study.
Sure. If anyone has questions.
Yeah, feel free to raise hand or unmute and come on camera. Carol.
Nice summary, James. Thanks for that. I'm pretty sure you've done this analysis, so I was wondering if you could comment on it. What if you pull out functional mutations in liver cancer driver genes?
So let me just stop sharing this. So we also tried to do that, but over 5,000 out of 5,500 mutations were liver specific. And when we trim it down to just clonal expansion, there were around just 20 mutations that aren't liver specific. So it doesn't really make sense to remove them. And even if you remove them, there weren't any significant differences between punches and homogenates. We've also tried Barbara's median absolute deviation, and we didn't find any difference between treatment groups and between punches and homogenates. So it's been very tricky to arrive in a conclusion when we don't see much treatment effects. That's what I think.
Thanks.
Thanks, James. Thanks, Carol. I see Paul's hand next.
Yeah, so I mean, James, first of all, thank you for all of the tremendous amount of work. And my question is kind of leading off of how you just finished that last. So given the lack of treatment-related differences that we see, right, between this molecule that we would have expected to see a robust change compared to the vehicle control, for you personally, James, since you're, I think, the most familiar with this data set out of all of us at this point, what would be the rationale, either from a process step or from an experimental step, why we didn't see potentially what was expected from a either is it the liver cancer driver genes that were selected for the analysis, or like, do we need to expand on what's being analyzed? Just what were your thoughts?
So this is just my personal opinion. I think it was too early to be able to detect uh, clonal expansion. And I think that's also the reason why we tested benzobifluoranthine, because at 90 or 180 days, they saw large mutations, and in some of the tissues that they analyzed for MMP, the mutation panel, they saw some clonal expansion. So it could be a really good test case whether can we see at these at this age, can we see clonal expansion? But for this specific imidazolidine analog, I just think it might be too early.
I have a hypothesis too, and I think that's the most important thing we need to take into consideration. These are non-genotoxic compounds. And with the compound we're going to see next, that's a genotoxic compound, and they behave differently. So for a non-genotoxic compounds, in order for them to function, first of all, there are more random the effects. So it's hard to find them. They take longer to kind of getting to the point that you can form a mutation, and that I agree with James, time frame could be. So not finding them is not rare. Of course, the genes also we have not completely validated those cancer driver genes, but having a non-genotoxic compound makes a big difference. And when you see the next data set, that's when you will see the difference that you will get. And even the timing, James, I think in genotoxic, you will see something, because it's much more broader, the effect. You see it everywhere. Here is more random, and we don't know we're seeing it.
Yeah, I mean, I think I completely take that point, Patricia. I think that's a valid component. I think from our perspective and from many of our, within the group's perspective, is these are the kinds of, more subtle and more hard to detect events that we're trying to go for. So then if we think that for this sort of mechanism broadly, right, not specifically to this molecule, if we think that it's an issue with timing, right, but this is sort of the step in which we would like to apply this technology or this sort of analysis, then do we need to talk about changing the depth or the types of sampling then that go into in order to ensure that, there's confidence in and potentially detecting something that could take longer to become more obvious, because I think that's the entire purpose of this technology.
Yeah, and that's the challenge we have, especially because we have not designed a study for that purpose of the technology. We're using samples we have available. So
right,
that
no, of course, absolutely.
And I agree, if we need more time, we need to do a time course to really understand where it comes. I've done a little bit more in depth when we did a study, PCB 126 in rats, where we didn't see much, we did more in depth. And we got a little more, but not that much more. Although right now the depth that you have for these non-genotoxic compounds is really high, much, much higher than you do with a genotoxic compound. So that's taken into consideration. But I agree with you, that's the challenge we have. Think about it, this is kind of the second study ever to be done with this type of compounds. So I don't want to discard the technology just yet. I think it's that we need to think a little more if we want to look into, and that's why it's challenging. We know the non-genotoxic compounds is not the easy one. So, but agree. I think we have a couple of other people, but that understand. So that's kind of things that we can think about it for later when we start discussing what's next.
No, absolutely. Thank you both.
Francesco.
I mean, just to add that, yes, in addition to that, the timing may have been too early. Another factor to keep in mind is that the carcinogenic effect was weak. So, 20% so of the animals developed cancer. So that also has an impact on how strong of a signal you can see this early.
Yeah, but normal non-genotoxic compounds are that weak. They're not that strong too. That's also a challenge.
Yeah, no, but again, it's the challenge is the question is more than the technology, I guess.
Yeah, agree. Okay, Megan?
Yes, I was just going to pretty much say what Francesco said, that, we are dealing with a non-genotoxic carcinogen that had somewhat, weaker carcinogenic signal, even though there is the hyperplasia, et cetera. So yes, it's likely it can be timing, but it could also be compound as well. We just don't know. But where was I going to go with this? Yeah, that was the main thing. Oh, the depth that Paul brought up. And the other thing to consider is if there is truly a strong clonal expansion signal, increasing the depth is not going to allow you just to suddenly see it. So if something's at very little fractions of 10 to the minus 4s, 10 to the minus 3s, 10 to the minus 2s, you're going to see it at a lower depth. When you increase the depth, you're just going to start getting additional alt depth counts of 1, and 1s move into 2s, and 2s move into 3s. So going from something of a 30,000 duplex depth to a 50,000 duplex depth or 60,000 duplex depth, you're not going to just suddenly see something at a high variant a little fraction that you wouldn't have seen at a lower depth. It would be observable at a lower depth. So that's all for now.
Thanks, Megan. Naveed, I see your hand up next.
Yeah, thanks. I was just wondering how big the CDM panel is you're using. I just didn't get it. So how many different CDMs are you using?
Oh, sorry, I was muted. James, do you want me to
Yeah,
pull that up?
the cancer driver genes?
Yeah.
23.
23.
Oh, I'm
Okay.
sorry.
It's
Not to be pedantic, 27.
27 cancer driver genes. It's 64 kb in total.
I think it's 27. That's why it's lower, because if you see that graph, 3 are for TGRAS, and we don't have that in these animals. That's why it's a little lower. But yeah.
Have you tried? I mean, this is obviously you've done it, but I mean, to see what would happen if you would categorize the different genes based on their hallmarks and look at those hallmarks which are towards the end of the become prominent towards the end of a carcinogenic process, and we would focus on that. Would that make a difference in your assessments? I don't know. Let's say angiogenesis. Angiogenesis is more towards the it comes towards the later part of genesis. Would that affect your results?
I mean, James, you had talked about like liver specific. I don't think we've thought about temporal specific. So that might be, if easy to label. I don't know if that's the hard part too, if we decide gene by gene where it comes. I don't know, James, do you want to start to answer that?
Just to help James a little bit.
Yeah, sure.
These cancer driver genes, when we chose them, it was based almost on some studies we did that was that TGRAS, and then others was based on data available from Riva, that's NIHS, and from actually Barbara Parson. So we assumed about those cancer driver genes already. So I don't know, Naveed, I'm not sure what you're asking. If you want to kind of go into one of each of them and see how they behave individually and understand if they have any assumption, because the assumptions when we chose this is that they had some type of activity in liver carcinogenesis. So I don't understand if that's what you mean, like kind of look into each gene itself right now with the data we have and see if it makes a difference, or if we have to question the type of genes that we found.
It's actually both. So basically, what you've selected a panel of different genes which are responsible. They're cancer driving genes. That these are responsible for the initiation and the promotion of cancer. Okay, and they belong to different hallmarks which are responsible. So I don't know, tumor suppression or oncogenic effects, and certain hallmarks are predominant, or they occur quite often in many of this, in many cells quite early. So that's why you're seeing these signals also in the controls. Okay, so but some come later in a cancer development, and maybe if we could just categorize the genes you're looking at to see which ones which ones would be expected to show up later in the development of cancer, and then focus on those genes and see whether you still do not see a difference between the two.
Okay, I got it. Kind of do a temporal space with all of the genes and see
exactly.
where is it, where genes will activate first and we activate later, and see if there's any trends there specifically to a gene. Yeah, okay, I got it. I don't know, James, what do you think about it? You're the one who have to think about it.
I think it makes sense for sure, and that's really very, very helpful strategy. But I would need the help of the experts here to determine those genes. I'm not really the authority to figure out those genes, I guess. But I think it's a good strategy moving forward to strategize the genes. We've tried strategizing it based on organ specific mutations or clonal expansion, and I think this is also a good idea to stratify it based on the temporal variation of gene responses. So yeah, I think that's a good way to do it.
So James, what we could do, we
Hey,
have
James.
we have participants of
Oh,
these
sorry.
people in the group, so later we can just kind of have conversations with them and see if they can help us with that. Yep. Go ahead, Megan.
Yeah, I've done a little bit of this work myself. And so like TP53 is generally considered a little bit later event than earlier, et cetera. I would say would be nice to do this. I don't know if this data set itself, because I had kind of looked at it in that aspect, is the best, unfortunately. But we could probably try to do a more stringent look for temporal changes. But I will just warn everybody that I don't think this data set ended up being the best for that.
But it might help us like going forward, or maybe a sub-analysis, right, Megan and Patricia, if like we could say, okay, if we're looking at a 90-day sample, maybe these are the genes you would focus on. That would be like a grand goal, right? Later, if we see this, or if you're looking at 180-day, maybe these are the ones.
Yes, so there's a series of events that's thought to occur. A certain mutation occurs generally early, and another one may be a secondary, and then the third one is like the real banger that causes finally the cell to actually truly start expanding and turn into cancer. That analysis can be done. I'm just saying that this data set's probably not the best one.
Carol, I think you put a comment.
I think she emphasized.
Yeah, no, I was just basically typing what Megan was saying while she was saying it, I realized.
But if there's others, like I know we're going to talk about BBF and that's more gene tox, but if, with those, if there are different time points, which I know they're available and we haven't done, maybe that's when we could think about this temporal nature.
I think at the end, like, again, we have to think about what is the purpose, because really we wanted to concentrate on genotoxic compounds because that's the challenge. But we can think about it if we can get proof of concept, as we tried to do with the next study, to see if it worked, and then think about it in this way, and then try to apply it into the genotoxic. That could be.
Right. And with the BBF, that's the one good thing about that when we did this study. It does appear that there is an induction and clonal expansion of hotspot KRAS mutations, which are the prominent early, basically you have two types of driven lung tumors. You have those that are driven by KRAS mutations and those that are driven mostly by EGFR mutations. And with the BBF, we have what I will argue is a true induction and clonal expansion of hotspot KRAS codon 12 mutations with dose.
Okay, so let's have a Carol, and then we can go to the BBF data so people can see it. Go ahead, Carol.
You muted, Carol.
I was just going to finish with the point of this was punches versus homogenates. And we don't see a difference in punches versus homogenates. So it's been successful in that sense. And I'm assuming then that we can move forward with homogenates because that would be cheaper. Does the group agree?
I do. Let's say who doesn't agree, because that's a little agree. So who doesn't agree, you can raise their hand and let us know. Okay, Paige. Go ahead. If you want to comment why you don't agree.
No, can you hear me?
Yeah.
No.
All right. No, I agree that punches are not really necessary. Because
Okay.
really, it's like taking a piece of the galaxy and trying to find a star there, and there's more normal space than not. I don't know. It just doesn't seem like we've done very well with punches finding the jackpot. That's all.
Thank you. Francesco?
So Jesse is probably not connected, right?
No, he's not here today, yeah.
And so he was the proponent of the punches versus the homogenate, which is something that I still don't understand the point, okay? But he had a point that he wanted to try. And I think to say that we have shown that punches or homogenates doesn't make a difference, it's a little bit, I don't know if you can make that conclusions because we are in a situation where there was no effect.
Yeah. So one of the things that he wanted to say is by doing punches as a non-genotoxic compounds, that the effects are random. By random mean having punches, you could maybe for a probability increase the chance of getting that small focus area. Doesn't mean it worked, but that's the principle that I recall. And Andrew?
I agree with Francesco. I don't think this is the data set. I don't think it really showed anything, but there again, I don't know if this exercise is meaningful or not. So
Thank you, Andrew. One thing that it could be meaningful is at least it was not that different, because what we can do is in the future, or do we repeat this when we have a study that we can repeat it, that we think is going to be positive, or we just assume, just start with the assumption that by default, if you homogenate, you have it there. I don't know. Anyway, we can discuss that later. Thank you, Carol. I want to give chance to James to finish the other study, and we can have this one in mind.
Right. The second one will be much shorter. I won't be showing lots of results for the BBF yet. Slideshow. So here is our second study using benzobiflorantine as a model, mainly to validate the statistical models and help establish the data analysis pipeline. And the rationale to using BBF is that while it's a genotoxic PAH, and we do acknowledge that there might be different mechanisms between this genotoxic compound and non-genotoxic compounds. BBF also has a CAR and AHR activity. So in addition to that, the samples from this study have been thoroughly tested by other groups. That means we can really leverage those information to validate our results. So as a study led by Stephanie Smith-Rowe from NIHS showed that the BBF 180-day lung samples were positive in the duplex-seq mutagenesis panel. And the data from Barbara Parsons' group from FDA NCTR who did CARC-seq also suggest clonal expansion. So these positive findings really have led us to test concordance between those positive results with duplex-seq carcinogenesis panel, our panel that is asking the question, will we be able to observe similar levels of mutations or patterns of mutations? So for this study, we used one of HESI's remaining duplex-seq kits for the 180-day study. And depending on the results of this 180-day study, we can consider using a second kit to process the 90-day samples if the signals for the 180 days is really high. So here's the schematic experimental design. We had 4 treatment groups, including the controls with 6 replicates each, where we've processed the 180-day sample, lung samples for now. The libraries were prepared here at U Ottawa from the lung DNA sent to us from FDA and CTR. The libraries were then sequenced very deeply by Merck, and we are currently working on data analysis. So here's a high-level look at the preliminary bubble plot results. We can already see an increasing trend in the number of mutations with concentration. There aren't really too many large clones that we've seen, and the pattern is quite similar to what Stephanie saw in the mutagenesis panel. So the data analysis team is now digging deeper into the clonal expansion metrics. I think the next big question, and really the main challenge here, is reaching a consensus on how do we define clonal expansion and how do we measure it. So I won't be showing other results for this one. We're still working on that. So I'll pass it back to Connie and Patricia.
And we decided not to, meaning the data is still not complete. And I know that James have done a lot of work. So we felt that it would be best to have like a complete story. We'll bring back this group to present it, because it's to give actually James the opportunity to show all of it, all the details. So I just want people to ensure that. But I think it would be good if you have questions already of anything that you want. Maybe he doesn't have the answers, but maybe it will help in the analysis. So we can open it up for questions.
Yeah, any thoughts? I can just share the conclusion slide. Any questions on this study or kind of ongoing plans?
I think what it would be interesting here eventually to know is like why you see clones. Is there any particular type of clone that you see over time, like one of these genes are better than others? Although one thing also is that we use a panel was designed for liver, and we using it in lung. So it's something else to take into consideration. But at least you're seeing the increases in general.
Yeah, Andrew wrote, love the bubble plots. I agree. I think these are really easy just to see what's going on. So I don't know, James, if you came up with these or whoever did, but they're really nice.
It's a Matt Meyer's group that we used for constructing all of these. So a shout out to Matt and Annette for building Mute Seeker.
Okay.
Oh, awesome. And Matt put the great.
Put it in okay. So if we want to go to the next slide, if there's no questions, feel free to ask James if you guys think of even ideas of how to do the data analysis. I think this is the right opportunity if anybody has any thoughts.
Yeah, so we just have a few more slides. Oh, Rick, you have your hand up. Go ahead, Rick. And you're muted if you're trying to talk, Rick. You're still muted. Oh, there, you're unmuted now. Rick.
Okay. Yeah. Just a quick question. So are those real mutations in the control? And the pattern looks interestingly similar to the other samples. Just so what I'm asking is there a low level of mutations that are just being expanded by the compound?
My short answer is possibly. We haven't fully cleaned up this data set. I haven't really looked closer, but what I observed here is that there are very small clone. I don't want to call them clones because a lot of these mutations, they only have one alternative allele depth. And whether we call them small clones or not, I don't know. I think that's a discussion for the whole group, whether we should consider those as clone. But I could only say for now that there are increasing number of clones as the concentration increases. for the control, I think I should say that we should still try to clean up and look closer at these bigger clones that we could see here at control group.
Yeah, thanks, James. Megan, do you want to add to that?
Yeah, so this becomes quite a bit of a fuzzy zone. I mean, we're dealing with a genotoxic carcinogen. We have clear induction of mutagenesis and increased mutation frequency with dose. So with dealing with a non-genotoxic carcinogen or all the published papers that I'm aware of with duplex sequencing, anything with an alt depth of 1 or more is somewhat de facto considered clonally expanded mutation. Here, with dealing with the genotoxic carcinogen, it is a bit fuzzy. There is an argument, I would say, that an alt depth of 1 or more could be considered still clonal expansion when I brought up about the KRAS mutations before, because these are occurring in that context of that codon context more than what would be probable by random mutagenesis alone. So I think this is something that we're going to need to do a lot of work on, and better statistical analysis and arguments back and forth on what to consider clonal expansion and not. So it's going to be difficult. And so far as I'm aware, there's been no threshold of 2 or more to call a clonal expansion. I know with Patricia's and Donna's, their work with the PC, was it PCP 126? I can't ever remember compound names. That they plan on having published where it's 2 or more to consider it being clonal expanded. But looking at this BBF data and looking at the looking at the KRAS mutations, there might be an argument for at least known hotspot driver mutations, specific codon mutations, to be able to consider being actual induction and clonal expansion, even at an alt depth of 1. Just throwing that out there.
Okay, so thank you, James. This is only just throwing out there for you to see, have ideas. So I think Paul had a question.
Yeah, in the chat, Paul asked, are any of these data outlier? Thinking of the large induction in the control, probably this one, right?
Yeah, there's that large bubble. And then
Could
how we
be.
think about outlier detection across the treated samples, right?
Yeah, something to think about.
Yeah, I agree. It could be an outlier, but then if you think about it, if there are some clones occurring in 1 sample or 2, it could be that specific clone it could be that with the jackpot tissue, I guess, or I don't know. Do we consider them as an outlier, or do we still keep them? Do we expect every single sample to have the same clone, or if they showed up in 1 sample, do we consider it as an outlier? So I don't know.
No, I agree. I agree for the treated ones. I definitely wouldn't use outlier because, like you said, any of them could be the jackpot sample, right, where you're trying to define and identify these mutations. But when it comes to the controls, I think that might be a different conversation. But
it's true, right? Because
similar
controls will
arguments.
still go to get tumors sometimes,
Of
right?
course.
Yeah.
Right. Yeah. So maybe we don't use any outliers then.
We'll see. Well, thank you, everybody. I think this is great. Thank you, James, for presenting. Carol, I think you have one question.
Yeah, I just wanted to ask one more question because I don't know if you've done this yet, James. What is the mutation frequency in the cancer driver gene panel overall compared to the mouse mutagenesis panel? So now you have genes and coding regions exclusively, and some of them are, well, they're all very important genes, versus neutral loci, not under any selective pressure at all. Do you know how the MF mins compare?
They were around e to the -6, the highest, but mostly e to the -7 or e to the -8. They were very low.
For both, so it was lower than the mouse mutagenesis panel? Or you don't know yet?
I don't know yet. I
Okay.
do have the data, but I don't want to give conclusions on that yet. I
Okay.
want to review the data for now.
You've got a lot of mutations. So
Yeah.
I'm just thinking about mutagenesis analysis in these genes as well, right? So we've got.
Right. And that is one of the studies that I am currently focusing, comparing what we're seeing with the cancer driver gene mutation panel and the MMP, the mouse mutagenesis panel. And we got some initial data set on that, which kind of shows that they're very close, but that may be a conversation for our next update.
Okay.
We'll just leave everybody wanting to come to
Yes.
Megan, last question, and we can move to the next one. If you have anything you want to add.
Not a question. I was just going to piggyback on Carol's question. Is I think the CDM panel was just slightly higher than immunogenesis, but everybody needs to know that these are two different totally designed panels. And the mutagenesis panel was very carefully designed. And when you're looking at cancer driver mutations, you can't ignore certain problematic regions, et cetera. So it's not to be, it's not surprising at all that you would have a slightly higher background in the CDM panel versus the mutagenesis panel.
Thank you.
Thanks, Megan.
Megan.
All right. Huge round of applause for James for presenting and all the hard work you've done. This is really a ton of things condensed into like 20 slides, so thank you. We just have a little bit more on what we've done this year. That was part of our goal, was really to disseminate part of these results. So we had a couple in-person presentations or posters at meetings. So James was able to present the punches versus homogenate work at EMGS a few months ago in Rochester. Earlier this year, Patricia presented, I believe, on behalf of James at the GTA meeting that was in Delaware. We have a picture with her and Carol chairing there. And then I had a poster at Eurotox, so this was really nice to try and connect with some of our European members as well. So we had a couple different outreach there. This is, again, things we can think about kind of in our packaging for next year. How do we tell these stories? For where do we want to present and when? And then we had two really nice webinars over the summer, and huge thanks to the people who presented. I think at least most of you are here. So we had a really nice presentation from Matt Meyer, Josie, and Annette Dodge about Kodak. And so that was really more focused on mutagenesis, but got us thinking, is this something we could watch and see how we could use it down the line? And then also Alina from BASF and her colleagues presented about kappa hyperpete. And again, this was in the context of mutagenicity, but it really helped us get thinking of kind of what other technologies are on the horizon that we could potentially use. So I think that's what I have in terms of outreach. Patricia, do you want to move to 2026 plans and talk here?
Yeah, so part of what we want to use in this is kind of outline what we want to do next, and really for you guys to start brainstorming what do we want to do, and if they have any proposals. So again, as James mentioned, we're going to continue the data analysis in the two study data sets. And the idea is to make sure that we can put some thoughts about how the data is analyzed. I think that's something missing in this area. So that could be, even if it's negative, but just the thought process of how to analyze the data, how to clean the data, and how to present it. I think that will be one of the things that we can come up with that. And then try to come also with that, what we call the data analysis pipeline, to support anything that is coming next. And we want the group to be involved. We have a subgroup that is called the data analysis group that works there, but if anybody's interested, they're always welcome to come. And then the last one is we're trying to explore for new samples, and almost going back to what Paul was saying, because I think I agree in the principle that we're missing it, and it would be good to nice to know why we're missing it. And again, we have not decided to do studies yet. That's something that we could think about it for the future. So we were going to think a different avenue. So we're looking for, I think, in the agrochemical industry, they may have had samples that are longer than the 180 days. I think they do like a 1 year sample 1-year study, and then they have like a 2-year CARSA study. So we're trying to see if maybe we can get another set of data around that, especially for non-genotoxic carcinogens, and see if we can get a better understanding of that. So we're looking into that. If anybody knows of data sets about that you can share with us. We will not share structures. We will not share. The only thing we want is the principles that we started with. That we want at least in a 2-year we have or a tumor, or that would be better if we have a cancer, or if we have hyperplasia, very high hyperplasia, and maybe see what happens in 1 year. So that's something that we're thinking. And then the last one is just kind of discussions. Where does this group think we should go next? I wanted to open it up and have that discussion. So if you have any questions of the plans, or if you want to discuss anything next, please feel free to ask. Anybody? Go ahead, Carol.
Well, I'm going to put James on the spot again, because I do think, we have one other study that we've been doing here at the University of Ottawa that borrows samples from the carcinogenomics group in eSTAR. And I'm not sure people have heard about this yet, but it's another really interesting study where P53 knockout, I think it was rats. It was rats, right, Connie?
Yep.
Yeah, those are rats.
Okay, there's James. James, do you want to say what you did with that one? Because it's very relevant. I mean, these rats all die of cancer by 4 months. I was gonna say 4 years, 4 months of age. And so what did you do with that?
So first, the experimental design of the other group was that they exposed knockouts and wild type rats into chemicals. And so far, what we've done is to use the CDM panel to analyze if there are clonal expansion between the wild type and P53 KOs. And I didn't see too many mutations, actually, not even clonal expansion between wild type and P53 KO. So what we wanted to proceed now is to run the same analysis with treated mice, both for P53 KO and wild type.
So James, the mice die at 4 months of age. The knockouts die at 4 months of age of cancer, and these mice were 2 months of age. And you did the cancer driver gene mutation analysis on the controls only, and
Right.
the wild types and knockouts. But there's no evidence of any clonal expansion of any genes. So you need more than just P53 mutation. You're halfway there to getting cancer in the tissue, and yet you're still not seeing clonal expansion. So either the panel, or the approach we're using isn't getting us where we want to go.
So for example, for that TG-RAS study that we did with TG-JAS study that we did PCB 126, we saw some clones. We saw tumors at 6 months. We saw some clones at 3 months, but they were not very strong. So maybe you need to go to 5 months. Maybe you need to have that threshold because it's a non-genotoxic compound. Maybe you need to be very close, and maybe they grow up very much. I don't know. Maybe that's happening here. We're assuming because we have, 2 days is not a lot of difference, but
yeah.
Just,
anyway, I think it's interesting for people to think about that as well. We also thought that when you knock out P53 that you'd have higher mutation frequencies, higher mutagenesis. And
you didn't.
There's none.
So maybe what we could do is also present this data in a little more detail next time. I think it will be.
Definitely.
Yeah, thank you. Anthony, I think you have your hand up.
Yeah, I think I was following on from where Carol was going. I kind of wonder whether we're somewhat fishing in the wrong pool, and that if there are models like the transgenic models with increased tumor burden, much more reduced latency, then that's where we should be fishing to to develop the POC side of this. And I've been talking to Alex Kagan at the Sanger because of what they've been doing with human cancers and understanding clonality and cancer driver genes. And they're looking at laser capture microscopy and analyzing tissues at a cellular level as well. So I just wonder whether, yeah, we need to be a bit more forensic about this.
Do you mean when you say frantic is like design maybe a study and try?
Well, there's two aspects, I think. One is if we use materials from, say, TGR assays or indeed wild types where we know we've had high tumor burden and very low latency, this then, you know, we're doing 3-month studies and we're seeing tumor burden and we can hopefully see the changes that we require, or do follow-up experiments with time course that we could actually interrogate what's going on, rather than always have to rely on 2-year or 1-year studies, which is clearly prohibitive. And then I think the second piece possibly might be that is there a possibility to do some tech dev on archive tissues to see whether we could use those? Because the other piece of this is that prospectively generating data is expensive and therefore restrictive. But if we could access archive material where these kind of where tumor genesis has been observed, is that another route in? But of course, you know, nowadays we're using kind of fresh material, fresh frozen material, and we probably haven't gone into formalin fix. And I know there's going to be issues around that, but if we could solve that problem, maybe we open, you know, a treasure chest of samples with which to explore. Those are the thoughts I've been having.
Thank you, Anthony. So regarding the tissues, so one of the things that when we discussed this earlier with Jesse, and I know he's not in here, but he says that if you keep the tissues too long in formalin, the DNA is not very good when you have to sequence so low, like so deep. So, but we could do, if somebody has the samples on the tissues, maybe we can just even do that experiment. I know that eSTAR did it many years ago, even with the transcriptomics, where they were trying to do that. So what we need to understand, if people have the tissues or things like that, that they can help us, we can do that. And then the time course, one of the things that I was thinking of proposing, maybe we can partner this group, because of course, you know, TGR studies are expensive, but if we can get a lot of partners and volunteers, I know that Taconic will be more than happy to help us maybe to get some of the transgenic studies, conducting the studies. We could maybe design a study, a TGRAS H2 study, 6 months dosing, time frames, and think about it. That's kind of where you were going, Anthony, kind of do it from scratch.
Exploiting those kind of opportunities, I think.
Yeah.
And I think it's worth talking to Sanger, because I think when they're accessing human tissue and human cancer material, clearly they're using legacy samples. Now, you know, the limitations they've encountered, or the workarounds, is it not worth having a conversation? Because, you know, these studies are ongoing in humans, and maybe there are some lessons to be learned
from their technology. Okay.
Yeah.
So
yeah, I'll just speak on Patricia's behalf. I would say that yes, you know, Taconic could participate, but one of the biggest limiters, which is kind of where Connie and Patricia are coming from, is really on the compound side, right? We faced this with our original study, and you know, so it's really an engagement of this team in terms of what are the potential options to make an in-life study possible.
Thank you, Janelle. I didn't want to talk for Taconic, but thank you for chiming in. So why don't we let Anthony, if you could at least, let's start, if you can help us kind of see, we want to connect with Sanger at some point to understand what are the
of course
apotypical action. Maybe we can get that going, a small group, whoever wants to participate, and if you can help us, we can do that. And then what we could do is, why don't we, I will put it out there. Let's think theoretically. We want to do a TGRAS H2 study next year. A couple of things that are critical will be the compound, what compound we want. We think maybe we can get some of the animals, and then who wants to run it, and see we can get volunteers. So what we can get started with is maybe people may just kind of let Connie know, yes, I think it will be a good idea or not. And if we get a good number of people that think they can help a little bit, some people can help in the dosing, some people can help getting the compound, some people can help and I'm talking companies, I'm talking universities, I'm talking everybody that can help. Some people can help with can find a pathologist that can help us get some samples readouts or get some tissues collected. I wanted to put it out there. I didn't want to put it here. It's one thought I have always had, that it would be nice to have a well-designed study and we can try to answer more questions. But the feasibility of doing that, we don't have the money, that's for sure. But it will be all volunteer. So I want to hear thoughts about that, meaning would people can try to help? And maybe we can start putting ideas together around that.
Yeah, maybe Patricia, do you have a prioritized list? I heard time course obviously come out of this group. But like, what would be the questions, right, that we would want to answer with the in-life study would be probably the main thing that, you know, I would need to know. And for this group to help decide, right?
I think the issue would be compound. What compound do we want to try?
Absolutely. I can't, yeah, it all comes down to what do we even have available and what could the compound address in terms of questions. But I was just thinking even beyond that, very limiting steps. So,
yeah, thank you, Janelle. Okay, I know this is a big question, so I'm not expecting people to answer. So I'll start with one with at least with Anthony checking out, because if we can get a better way of getting tissues, maybe that open up new freezers for us. One, and then just keep in mind about this transgenic, and we'll see if we can think of something for next year. Megan? Go ahead.
No, I just wanted to reiterate that, you know, we have these pre-existing purchase kits that are sitting in a freezer, and our best approach, I think, immediate would be pre-existing tissues, just to relay to anybody if they have any existing tissues that we could really use them and to analyze for these kits, because I don't want them to sit there for several years, especially if we start talking about designing a study. So if anybody has anything available, even if it may not even be perfect, I think we'd be better off just to start working and analyzing, even if we have maybe lower expectations for a particular compound, and start building up our data set versus letting these kits sit forever in the freezer.
Yeah, so we did start the approach, Connie and I, and we're getting companies checking to see if they have, I don't want to say the name because they have not said yes yet.
Right.
But they're checking and see if they have those samples that I just mentioned, the 1 year with the correlation of the 2 year. They're looking into that, so we can get started with that. But please, if anybody has anything, and I know, Navid, I know you're working in some studies, if you have some there, and I don't want to put you in the spot, but we have some kits that we could potentially also help to use if you need. Thank you, Megan. Okay, so I think we captured a couple of actions or follow-ups for next year. I like that idea. Anything else from anybody?
Just,
go ahead.
just want to make the comment. We're still in the gestation phase, but once we get going, we will really help out. Okay, just give us a bit time.
No, we have some kits, so if you just to think about it, that we could use those kits. We do it actually. We have the TwinSat kits. I know you're using a different technology, but we could potentially use some of those if you need to. Okay, thank you, Navid.
What we're working on right now is, sorry, just for your information, FFP material is not a homogeneous quality tissue. Sometimes they're fixed for a week, sometimes they're fixed for 6 months, okay? And we really don't know what that has an impact on a retrospective analysis of what we're all talking about right now. And so basically right now our focus is, what is the impact of formalin fixation, the kinetics of formalin fixation, period, okay? So we're working on that right now, and this is I think it's very important for us to address before we start using them and produce data.
Yeah, no, thank you. I think I agree with you. That will be
And
awesome.
as soon as we have data results, obviously we'll share them with you.
Thank you, Naveed. That's awesome, actually. That's a very good exercise.
Sorry, no, it's wrong.
Okay. Thank you.
Yeah, we had some past work. I saw Leah earlier, but she might have had to go at the hour. But yeah, we have some publications on FFP, on like historical samples, like from museums. So happy to revisit those and see if there's any good learnings.
So yeah, could you share that with this group? Because I know that was done in that more in the.
Yeah, let me stop sharing it. And I'll get the papers.
Yeah, that'll be. Go ahead, Gary.
Go ahead, Deidre.
Hi, it's Deidre. So just in regards to, I would say, the formalin fixation, I think, as everybody was saying, a lot has to do with the speed at which those samples actually get into the formalin, for one, I would say. But I have a lot of experience in looking out to samples that have been fixed for very long periods of time. I would say out, past 3 years when, in my past experience, and I was unsure if they would work. A lot of it has to do with, what that downstream endpoint is. When it comes to, I would say, transcriptomics and genomics, I would say generally transcriptomics, it really depends on where those primers and probes are generated. So if they are down at the one end, you're still probably going to be fine. So with samples that are fixed, I've had ones that worked out to 3 years, and if I took our normal primers and probes that we used all the time and didn't care about generally, where that primer and probe were generated, just where the, say, applied biosystems, system, if you buy them off the shelf, where they were generated. But it's more about looking at where those primers and probes are located and optimizing them if there are a number of different ones, or generating ones that are in the right spot. So I think that even things fixed for a very long period of time still have the potential to work beautifully, as long as they got into formalin quickly and you have the ability to understand where those assays are located at. I can predict what would work based off of is it 5 prime, 3 prime in the middle? I have a lot of experience doing that. I'm happy to, have further conversations about it and explain what I would do in order to be able to use those samples that were, fixed and then archived for periods of time as well.
Okay, perfect. Thank you. I think we can put that. Thank you for offering to help us with the experience on that. Thank you.
Sure.
Okay, so let's put that one as one of the other topics for us to revisit for next year. Yeah. Okay, perfect. Thank you for everybody for the great feedback. James, for your great amount of work. Connie, for your guidance. Connie's the one who keep us going. So thank you very much, Connie, for your support and work. Anything else from anybody before we close? And I actually want to say appreciate all of you, because you guys show up the majority of our meetings, and I know it's not a lot of data, but at least we all have the idea that we want to make this work or understand. So really appreciate that. Okay, if we don't have anything else, Connie, you want to close that meeting?
Yeah, I just really appreciate everyone's attention. We'll send out the recording. Please flash freeze. Yeah, exactly. Maybe a methods ask for that. And just thank you all, and we'll continue with data analysis. And I think as Carol and James said, hope to present some of the, the rest of the BBF study, the knockout study, and some other work early next year. So thanks, everybody. I hope you have a great rest of your day, and thanks for all your hard work.
Okay. Thank you, everybody. Have a nice weekend.
Bye, everyone.